Hey, welcome to our teaching video on the synapse. So let's talk a little bit about the synapse. First of all, what is it? The synapse, first of all, you might hear other names on the test. It might be called a synaptic gap. It might be called a synaptic cleft. But nonetheless, what it is, it's the space between, or the gap that exists between two neurons, or the gap that exists between a neuron and a muscle. So if you see this particular case here, you can see here is one neuron, and here is the other neuron. The gap that exists in between, they've blown this up, that space becomes the synaptic gap. So that's the area that we're talking about. Now, why is this so important? What we've seen up to now is we've seen how an impulse has been propagated down the entire length. Sorry, let's go from this one. Down the entire length of a neuron. So this is the impulse. So we know that to be as I guess described as a wave of depolarization that occurs down a particular axon. So we've talked about this before. What we haven't shown you now, and this is why we're talking about the synapse, is how we now take that wave of excitement, that wave of depolarization from one axon and pass it on to the axon of another neuron. The reality is that you just don't have one sensory neuron going all the way to the central nervous system. You'll have thousands of these neurons. So what's really important is that we see the impulse going down from one neuron. How do we pass that wave of excitement or depolarization to the next neuron? Or how do we pass that impulse transmission from one neuron to the adjacent muscle in order to get that muscle to contract? So that's why synapse is so important. So if you take a look at your steps, this is on page 21 of your notes, topic 5, the synapse. What we're going to do is we're going to draw a couple of reference points, a couple of pictures here, and then we're going to go through those steps. Very important. You're going to see this on the exam, and often you're going to see this in the capacity of a numerical response where they're going to list a bunch of different examples of these steps, and you have to put them into the correct order. So let's just draw a couple of things here first of all. So let's draw, and then we can explain a couple of things that maybe you might have problems with when you were looking at your notes. We know that synapse occurs at the end of a presynaptic neuron. Presynaptic neuron. Okay? And we know at the end of that now pre means before. So before the actual gap, we call that the presynaptic neuron. Okay, and we also know that at the end of that neuron, we have axon terminals. And these terminals have vesicles, neurotransmitter vesicles. And I'm just going to shorten this because we want this video to be a little bit faster. And inside these vesicles, we have certain neurotransmitter chemicals. Okay, one of the examples that we're going to draw for in this picture is acetylcholine. It is the neurotransmitter chemical responsible for exciting a muscle. Okay, on the other end, let's just put a muscle. Now, if this was a neuron, we would call it a post-synaptic neuron, right? Because it is post meaning after the gap. Or in this case, we're going to call this a muscle. We want this muscle to contract. And on this muscle, or if it was a postsynaptic neuron, we would have structures called protein receptors. And those are found on the postsynaptic neuron or a muscle. Okay? This gap in between here now, we've already determined to be the synaptic gap. So that is the synapse. So let's go over some of these steps now. So the first thing it talks about, and again, these steps are directly off of topic 8 on page 28. But basically it says the impulse finds its way to the axon terminals. Wave of depolarization makes its way to the axon terminals. That's going to cause calcium uptake. So this calcium that's outside of here, it's actually 2+, is going to be taken up by the axon terminal. That's going to cause these vesicles to fuse with the membrane and dump their contents. And again, their contents are the neurotransmitter chemicals. And the one that we're going to use, because we're talking about a muscle here now, is acetylcholine. Okay, that's the example that we're going to use right there, acetylcholine. So calcium comes up. That causes the vesicle to fuse with the membrane and to dump its contents. And I've seen a question before that they talk about this process as being exocytosis, exiting the vesicles. And this actually needs some energy. We need some energy. We'll have mitochondria in there inside that presynaptic neuron that will actually provide the ATP energy to initiate this process. Okay, but that's not one of our steps. So let's go to number 3 now. These chemicals, neurotransmitter chemicals, in this case acetylcholine, will diffuse across the gap. And what they do is they dock onto these protein receptors. And it's kind of like a lock and key. If that actual key fits, so these chemicals are different shapes, and you have different protein receptors with different shapes on the postsynaptic neuron of the muscle as well. But when the right neurotransmitter chemical is released and it fits on the correctly shaped protein receptor, it causes a chain of events. So what happens? Once they dock on here like this, what happens is Na gates on the postsynaptic neuron, or in this case the muscle, will open. So Na gates open. And what happens to the sodium that's inside here? Well, it's going to enter and flood the postsynaptic neuron, or in this case, the muscle. And when sodium enters a muscle, or the postsynaptic neuron, that causes depolarization. And now what we've done is we've passed this wave of excitement, this impulse, from one presynaptic neuron across the gap, and we now excited either the postsynaptic neuron or in this case the muscle. And when the muscle is depolarized, it contracts. And we accomplish the contraction of the muscle. Last step. As long as these neurotransmitter chemicals stay within the synaptic gap, this muscle will stay contracted, or the postsynaptic neuron will stay depolarized. We know we don't want that. We want the muscle to contract, but it's just important to have that same muscle relax. If this is a heart muscle, we want the heart muscle to relax so it can fill with blood and then get another contraction to pump it. So the last step in the synaptic steps is that we must release, now your book talks about cholinesterase, but we often hear it called acetylcholinesterase. We have to release that chemical. And once we release that chemical, what that does, it clears the synaptic gap, including the ones that are docked on the protein receptors. It clears the gaps of all these neurotransmitter chemicals, in this case, acetylcholine. And once we do that, that will allow this postsynaptic neuron or the muscle to relax, repolarize, and get ready for another potential impulse to come along. Okay, again, very difficult concept. If you're struggling even after this teaching video, give me an email and let's book some time, either face-to-face, online, over the telephone, whatever it takes. But let's go over the steps, because you're going to see this question come up. And I encourage you to take a look at the next practice question with Synapse, where I give an example of how this question could be tested again on a unit exam, midterm, or a final. Okay, thanks guys, and good luck.